So you always want to consider your patient. What antibiotics might your doctor use? Another tip about the antibiotic discs, it is said, again, for general practice where you may not want to increase the cost for running a test, but tetracyclines and sulfonamides, sulfa drugs, only need to have one disc of choice because they often cross resistance is present. So again, if you had 10 different discs to choose from, you may want to choose one of those that would seem to be the best choice for your potential patient. You want to store these discs in the fridge, but they also don't like to be wet, okay? When not in use, so always read your directions. There may be specific applicators that the little disc tubes, the holders where the discs are in, applicators may hold these to actually apply them to a petri dish that you have, to a media plate. So if you were ordering, you want to make sure, and you have an applicator, that you are ordering the right tubes of discs that will fit your applicator. They may not all be cross-purposed. It is what they call indirect sensitivity testing. Requires a colony subculture that will be incubated, and then you create turbidity to match a standard, and that's where we have the term McFarland standard. So it is a set of glass tubes that have a turbidity changes, and you generally want to mix up your culture broth of your organism so that it matches 0.5 McFarland standard. And we will show you this again in the hands-on. But the McFarland standard represents colony count in a given suspension. Okay, so please reference the textbook on all of this. Again, you're going with this Kirby-Bauer technique. You're going to create a suspension to match that McFarland standard. So remember the terms MIC, minimum inhibitory concentration, McFarland standard. You have applicators, you have antibiotic discs. All of these actually make up part of the Kirby-Bauer antibiotic testing sensitivity test technique. You will then make a suspension and you apply that suspension with your organism, matching that McFarland standard, and you apply it to Mueller-Hinton media. That was mentioned in the PowerPoint 1. With that sterile swab, again, you want a rayon swab, not a cotton. And this is where we make a lawn. We call it like a golf course green. We want it really nice and even and smooth. So we will show this technique. Again, I'm hoping to illustrate that on a brief video to you. If you're using urine, you can just use that to make your lawn. And then you will apply your discs. And again, we will go over that application process. We use tweezers. We don't use an applicator at CNM. We don't have one. So you will be flaming your tweezers because you don't want any cross-contamination. You'll flame your tweezers according to the technique that we show you. Grab a disc, apply it gently onto the surface. You don't stick it into the media. It just stays on the surface. But it has to be applied tight enough to actually stay. You're going to incubate that plate for a constant period. Generally at 18 hours, we check it, maybe up to 24. Just depends a little bit how fast our growth comes. You might be able to perform an initial reading even at 6 to 8 hours. But again, this is also something where client communication is important. How come it takes, a day or so to get results, to know what medication to give the pet, and to explain to them it takes time to be able to actually grow these organisms out. Following the directions in the textbook and reading that area, you would actually measure around your disc where the clear area is, and not necessarily the greatest clear area is your best antibiotic of choice. That's why you have a table to actually refer to. You're going to measure it from the underside of your media plate, keeping it closed with a ruler, in millimeters, and then the zones are reported either as being resistant or susceptible, and you might have intermediate. They're partially susceptible. So again, that size alone is not the determining factor. Final initials to understand is CFU, for colony forming units. And that's again mentioned in the textbook, CFU. Some other tests that you will be introduced to. Read these up, and you will have a skill sheet next week to actually work on some of these in a written form. So please do read the textbook on these, because these testings, as you will see next week when we introduce organisms, different organisms may have a different test kind of result, and that is part of your work in your dichotomous key. That is part of how you determine a preliminary or potential diagnosis for an organism. So determining that genus of a pathogenic organism is often possible by using just staining and culture characteristics. And we term this presumptive or tentative or possible. It's kind of like looking at a radiograph and without a label, but you think that this is potentially a right lateral abdomen because of the other typical skills that we employ in radiology. The same way, you would come down and say, a presumptive guess on this organism is, a tentative identification is. If you say this is XYZ, it may not get full credit on a test. Again, that dichotomous key, that method of asking those yes-no questions, and then making wise decisions in the order of the tests that might help gain the best knowledge, the quickest knowledge to a potential organism. And we'll highlight that a little bit more in next week's decisions as we look at various organisms. So tests that you will be doing an activity with next week is potassium hydroxide, KOH. It can help you to know if something is gram positive or gram negative. Motility. So we come back to those flagellas that some bacteria have. There is a test that we can see if they are motile. Do they tumble like a top, or do they move across the slide? Flagella might be present with one flagella. You might have flagella on both ends or multiple around. And all of those help with the motility of that bacteria. Catalase positive, a hydrogen peroxide test. Every office has hydrogen peroxide around, so it's a very quick and easy test to do. Oxidase, indole, and coagulase. These are three other tests could be utilized, and then subculturing off to a different kind of media. You'll be, again, exploring those tests next week. California mastitis test, also known as CMT, in your VT 1011 course. This was mentioned a little bit, but mastitis can actually be caused by bacteria or mycotic or fungal kind of organisms. This is the CMT is actually a qualitative screening test. We're looking for somatic blood cell presence, white blood cells, in the milk that would be a potential indication of an infection going on. It's also very important that with the petal, as illustrated here, has 4 compartments, and you'll see in your textbook also, that you always get the samples into the same compartment. Set up a routine if you have the handle going to the tail of your cow or goat, or if you have the handle going to your nose, whichever way you want, because the udder actually has divisions in it, in the tissue, that one compartment, 1/4, is not connected to the other quarters. So potentially you might be able to milk out and use milk from one, but not in the others. So it's important to always know which of these teeth you're getting the positive results from. So consistent technique, track your results. If it is being a routine testing of a herd, there will be a usual pattern, how often everyone is being tested. And it is an agglutination test. A gel is sort of formed instead of being a liquid, and that gel, that agglutination, and it's rated on a scale, actually will tell you that you have a positive result. Coming into our fungi or mycology, so we're going backward into textbook just a little bit, and one also at the end chapter, so briefly page 238, but also 273 to 277. Fungi are what's called heterotrophs. They can either be what's termed parasitic or saprophytic. So parasitic would be that they're actually taking nutrients, let's say, from your host. Saprophytic, they're kind of living in conjunction with that host and may not be as harmful to the host, though you still may want to get rid of them. Most of these are multicellular, except our yeast, they're singular cells. They contain eukaryotic cells versus prokaryotic, which prokaryotic cells are bacteria, but eukaryotic cells have cell walls that are composed of chitin. And most of the time, your fungi are going to create webs of hyphae and so forth. And they digest food externally through a release of digestive enzymes. They are actually broken apart by their reproductive systems. So they may have a reproductive structure that's called a fruiting body, which produces and releases spores. So different than how bacteria grow, fungi typically will have spores as part of their reproductive process. And different groups of fungi produce different types of spores. Yeast actually reproduce by budding, a budding system rather than spore formation. And again, most fungi rely on both sexual and asexual reproductive systems. So depending what's present may depend on how they actually will reproduce. So the sexual spores are ascospores, basidiospores, and zygospores. And we have 4 groups of pathogenic fungi based on these reproductive structures: Basidiomycetes, Ascomycetes, and Zygomycetes, and Deuteromycetes. You can read about these different ones. Okay, so looking at so we'll come to, that's about as much as we'll cover there on fungi. But do read the textbook and understand how they actually reproduce, and that there are differences. Then we come to our cutaneous mycotic organisms. This is ringworm, okay? Dermatophytes. You need to know that the term dermatophytes often is referred to as ringworm due to the circular lesion that is formed. And it's actually, these dermatophytes are classified by the habitat where they're found. So anthropophilic is only in people, zoophilic, animals spread it to people, and geophilic might be a type of organism in the soil. Okay, Microsporum gypsian. The 2 Microsporum organisms that we see primarily in our animals are Microsporum canis and Microsporum gypsian. We also see Trichophyton, Mentogrophides. And most of the dermatophytes grow on the outside of the hair shaft. So for the collection of these, you want to use forceps and swabs. Again, we will show you the technique, and there may be a video link that I can show you also on this. New methylene blue is typically going to be your stain eventually. Our culture media is called DTM, Dermatophyte Test Media, DTM, Dermatophyte Test Media, or it's also termed soberod agar. And in the presence of fungi, that agar changes to red. You might use a woods light to be able to actually help with some identification. And you may, in practice, might want to have a different incubator just for your fungi. That woods lamp would actually turn, look, is an ultraviolet light, and it fluoresces green if it's positive. So the DTM, your dermatophyte test media, actually has that indicator in it that turns red in the presence of most dermatophytes. Okay, unfortunately, not all of them have read that news. So once in a while, he'll have a dermatophyte that doesn't turn red, but most of the time it does. And generally speaking, within about 7 days, maybe even quicker, depending how hot of an infection it is. Although we will keep a dermatophyte test media around typically for up to 3 weeks until we actually term it, maybe even 4 weeks till we term it totally negative. You're going to again collect your specimen. You don't want to again use alcohol or anything. If it's very dirty, just a light water or just a little cotton brushing off, and you get the sample from the lesion area, from the periphery. The test tube itself, because these grow on the surface, you want them to have access to air, so you will never totally close or tighten down all the way the lid of this media. Okay, incubated at room temperature because it's on the surface of the animal. It does not need to be in an incubator, although you may have one that is at room temperature to just keep it out of the airflow. This is a slide that shows a little bit about how the organisms look, how the media changes color. It was a yellowish orange, it goes to red. And you can see the textbook pictures also. For yeasts, okay, generally yeasts are going to be larger than our bacteria, but the smallest yeasts are not so large as the largest bacteria. So again, once you start looking under the microscope and you get an idea of sizes, that will help you with this. Oftentimes yeast, and this is sort of the bold part here, are often egg-shaped. They have no flagella, no other organs of locomotion. Gram stain will often show yeast, although a stain that's called Sudan 3 may also help. New methylene blue might be utilized. And again, their reproduction is by budding, fission, okay, that kind of area. And yeast often attack our skin and respiratory or the intestinal tract. We have 4 different species. These are broken apart in the next slide. You can see on this one where they are primarily seen. I will not actually read all that over. You can pick that up on your own as we look at these. So blastomyces dermatitis, doesn't that make it sound like it's going to be on the skin? So indeed, that is quite where it's at. Inhaling the spore-laden dust may get you a blastomyces infection. Candida albicans, quite often in the mucosal surfaces, either in the oral area, can cause thrush or vaginitis, reservoirs man, and again, contact mucosal areas. Coccidioides immitis. Coccidioides immitis. You need to know that its alternative name is valley fever. Okay, the reservoirs and soil here around rodent burrows, and again, inhaling the spores from the dust, and you can see which animals and man that it will actually infect. Coccidioides immitis, valley fever. Cryptococcus neoformans, blastomycosis, oftentimes in spinal fluid, and a reservoir here is old pigeon nests. So again, if an animal has been around an area with a lot of old pigeon nests and they come up with a odd infection, this might be one of the yeast kind of infections that you would be considered, you or your veterinarian. Histoplasmosis capsulatum. Histoplasmosis capsulatum. Mold in the soil, and the yeast in animals, reservoirs, soil, especially around chicken houses. So chicken houses, caves, starling roosts might have this one. And histoplasmosis occurs in dogs, cats, rats, skunks. And you have some illustrations of how it looks. And our last slide here on this is virology. This goes back again into pages 239 and 240. You ended up studying several virus diseases, so viruses are not really tied into microbiology itself. Virology would be its own study, but a test question could be that viruses contain DNA or RNA, but not both. And we've been hearing a lot about viruses in the last several weeks. And indeed, the coronavirus is an RNA-based virus, and so viruses contain one or the other. A lot of times these will include histopathologic and serologic kind of examinations. Electron microscopy is necessary to actually visualize a virus. Again, if you think you're turning something in for virology, always check with your lab. How do you want that turned in? The in-house antibody tests are what we use quite often for like parvo tests, FIV, FIP, FELV, all those are viruses, and those are in-house antibody type tests. You want to always read the directions step by step. Does it need to be in the fridge? Is it at room temp for storage? What should your specimen be, etc. You might have secondary bacterial invaders that can actually complicate your viral diseases. Again, you're going to generally refrigerate your samples, but when in doubt, check with your laboratory if you're turning something into them. So cell cultures and serologic procedures are also available. So this completes PowerPoint number 2. This is the 2 sessions for the basis of week number 1. And enjoy studying. Remember to look at those bold items in the PowerPoint and some of the areas that I tried to mention. Read your textbook and enjoy. Thank you very much.